The eukaryotic cell cycle is regulated by a molecular control system. The timing and rate of cell division in different parts of an animal or a plant are crucial for normal growth, development, and maintenance. The frequency of cell division varies with cell type. That is, some human cells divide frequently throughout life, like your skin cells. Other human cells have the ability to divide, but keep it in reserve, like things like the liver cells. Mature nerve and muscle cells do not appear to divide at all after maturity. Investigation of the molecular mechanisms in regulating these differences provide important insights into the operation of normal cells and may also explain how cancer cells escape these controls. The cell cycle appears to be driven by specific chemical signals present in the cytoplasm. Some of the initial evidence for this hypothesis came from experiments in which cultured mammalian cells at different phases of the cell cycle were fused to form a single cell with two nuclei. Fusion of an S-phase cell and a G1 phase cell induces the G1 nucleus to start S phase. This process suggests that signaling molecules present in the S phase nucleus stimulated the fused cell. Fusion of a cell in mitosis in the M phase with one in interphase, even G1 phase, induces the second cell to enter mitosis. The sequential events of the cell cycle are directed by a distinct cell cycle control system. Cyclically operating molecules trigger and coordinate key events in the cell cycle. The control cycle has a built-in clock, but is also regulated by external adjustments and internal controls. A checkpoint in the cell cycle is a control point where stop and go-ahead signals regulate the cycle. The signals are transmitted within the cell by signal transduction pathways. Animal cells generally have built-in stop signals that halt the cell cycle at checkpoints until they are overridden by go-ahead signals. Many signals registered at checkpoints come from cellular surveillance mechanisms. These mechanisms indicate whether key cellular processes have been completed correctly. Checkpoints also register signals from outside the cell. Three major checkpoints are found in the G1, G2, and M phases, here indicated by the red blocks. If the cell does not receive a go-ahead signal, the cell exits the cell cycle and switches to a non-dividing phase, the G0 phase, G sub 0. Most cells in the human body are in the G0 phase. Liver cells can be called back to the cell cycle by external cues such as growth factors released during injury. Highly specialized nerve and muscle cells never divide. The cell cycle clock is regulated by molecules called cyclins and cyclin-dependent kinases. Rhythmic fluctuations in the abundance and activity of the cell cycle control molecules pace the events of the cell cycle. These regulatory molecules are mainly proteins of two types, protein kinases and cyclins. Protein kinases are enzymes that activate or inactivate other proteins by phosphorylating them. Particular protein kinases give the go-ahead signals at the G1 and G2 checkpoints. The kinases that drive the cell cycle are present at constant concentrations, but require the attachment of a second protein, a cyclin, to become activated. Levels of cyclin fluctuate cyclically. Because of the requirements for binding of a cyclin, the kinases are called cyclin-dependent kinases, or CDKs. Peaks in the activity of one cyclin CDK complex, the MPF, or the maturation protein factor, corresponds to peaks in cyclin concentration. I'm sorry, that's maturation promoting factor. Cyclin levels rise sharply during the S and G2 phases and then falls abruptly during mitosis. The MPF, the maturation promoting factor, or M phase promoting factor, triggers the cell's passage past the G2 checkpoint to the M phase. MPF promotes mitosis by phosphorylating a variety of proteins. MPF acts both directly as a kinase and indirectly by activating other kinases. MPF also stimulates fragmentation of the nuclear envelope by phosphorylation of various proteins of the nuclear lamina during prometaphase of mitosis. MPF also contributes to the molecular events required for chromosome chromosome condensation and spindle formation during prophase. MPF triggers the breakdown of cyclin, reducing cyclin and MPF levels during mitosis and in activating MPF. The non-cyclin part of MPF, the CDK, persists in the cell in inactive form until it associates with the new cyclin molecules synthesized during the S and G2 phases of the next round of cyclin. At least 3 CDK proteins and several cyclins regulate the G1 checkpoint. Fluctuating activities of different cyclin CDK complexes are of major importance in controlling all the stages of the cell cycle. There are internal and external cues that help regulate the cell cycles. Research scientists are currently working out pathways that link signals originating inside or outside the cell with responses by cyclin-dependent kinases and other proteins. The M-phase checkpoint ensures that the kinetochores of all the chromosomes are properly attached to the spindle at the metaphase plate before anaphase. In this case, the regulatory molecule is not a cyclin CDK complex, but a complex made up of several proteins. Once activated, the complex sets off a chain of molecular events that activates the separase enzyme, which cleaves the cohesins and allows the sister chromatids to separate. This ensures that daughter cells do not end up with missing or extra chromosomes. A variety of external chemical and physical factors can influence cell division. For example, cells fail to divide if essential medium is left out of the culture. Particularly important for mammalian cells are growth factors, which are proteins released by certain cells that stimulate other cells to divide. To date, researchers have discovered at least 50 different growth factors. Different cell types respond specifically to certain growth factors or combination of growth factors. Platelet-derived growth factors, PDGFs, produced by blood platelets, are required for the division of fibroblasts in culture. Fibroblasts, which are a type of connective tissue cell, have PDGF receptors on their plasma membranes. PDGF molecules bind to these receptor tyrosine kinases, triggering a signal transduction pathway that allows cells to pass the G1 checkpoint and divide. In a living organism, platelets release PDGF in the vicinity of an injury. The resulting proliferation of the fibroblast heals the wound. The effect of an external physical factor on cell division can be seen in density-dependent inhibition of cell growth. Cultured cells normally divide until they form a single layer on the inner surface of the culture container. If a gap is created, the cells will grow to fill the gap. The binding of a cell surface protein to its counterpart on an adjoining cell sends a growth inhibiting signal to both cells, preventing them from moving forward in the cell cycle, even in the presence of growth factors. Most animal cells also exhibit anchorage dependence for cell division. That is, to divide, the cells must be anchored to a substrate, typically the extracellular matrix of a tissue. Experiments suggest that, like cell density, anchorage dependency is signaled to the cell cycle control system via pathways involving plasma membrane proteins and elements of the cytoskeleton linked to them. Cancer cells, in contrast, exhibit neither density-dependent inhibition nor anchorage dependence. Cancer cells, in fact, have escaped the cell cycle control system. Cancer cells divide excessively and invade other tissues because they are free of the body's control mechanisms. Cancer cells do not stop dividing when growth factors are depleted. This is because a cancer cell manufactures its own growth factors. It has an abnormality in the signaling pathway or has an abnormal cell cycle control system. The underlying basis of these abnormalities is one or more genetic changes that alter the function of the cancer cell's protein products, resulting in faulty cell cycle control. If and when cancer cells stop dividing, they do so at random points, not at the normal checkpoints in the cell cycle. Cancer cells may divide indefinitely if they have a continuous supply of nutrients. HeLa cells, Henrietta Lacks ovarian cancer cells, have been constantly dividing since 1951. In contrast, nearly all mammalian cells divide 20 to 50 times under culture conditions before they stop, age, and die. Cancer cells appear to be immortal. Cells in culture that acquire the ability to divide indefinitely are said to have undergone transformation. Transformation is the process that causes cells to behave like cancer cells. Normally, the immune system recognizes and destroys transformed cells. Cells that evade destruction proliferate to form a tumor, a mass of abnormal cells. If the abnormal cells remain at the originating site, the lump is called a benign tumor. Most benign tumors do not cause serious problems and can be fully removed by surgery. A malignant tumor includes cells whose genetic and cellular changes enable them to spread to new tissues and impair the functions of one or more organs. An individual with a malignant tumor is said to have cancer. Cancer cells are abnormal in many ways. Cancer cells may have an unusual number of chromosomes, their metabolism may be disabled, and they may cease to function in any constructive way. Cancer cells may secrete molecules that cause blood vessels to grow towards the tumor in a process called angiogenesis. In addition to chromosomal and metabolic abnormalities, cancer cells often lose their attachment to nearby cells and are carried by the blood and lymph system to other tissues and start more tissues in an event called metastasis. Treatments for metastasizing cancers include high-energy radiation and chemotherapy with toxic drugs. These treatments target actively dividing cells. Chemotherapeutic drugs interfere with specific steps in the cell cycle. For example, Taxol prevents microtubule depolymerization, preventing cells from proceeding past metaphase. The side effects of chemotherapy are due to the drug's effect on normal cells. For example, nausea results from chemotherapy's effects on intestinal cells. Hair loss results from its effect on hair follicle cells. And susceptibility to infection results from its effect on immune system cells. Researchers now know a great deal about cell signaling pathways and how their malfunction is involved in the development of cancer through effects on the cell cycle. Coupled with new molecular techniques such as the ability to rapidly sequence DNA of cells in a particular tumor, medical treatments for cancer are beginning to become more and more personalized to each patient's tumor. The development of cancer is a very complex process with many areas that remain to be explored. Perhaps the reason there are so many unanswered questions about cancer cells is that there is still so much to learn about the functioning of normal cells.